Congress in 2023 is going to be in Rio, you got to be nice to Gustavo because you may need it.
Thank you, Vicky. Good morning, everybody. It's a pleasure to be here and talk about this work, Phylogenetics of Baccharis and Megadiverse Genus of Compositae. This work was made in the Universidade de São Paulo, Embrapa, that's the Brazilian Agricultural Research Public Corporation, and Gothenburg University in Sweden. First of all, I would like to say that when I was a child, I was born and grew up in southern Brazil, and it was a dream as a child to dig a hole in the ground and just exit in the other side of the world in China. Then I realized that it was not possible to do that, but I have to acknowledge Embrapa for letting me to come here, and the IBC with the Excellent Scholar Award that for the 5 tickets, and also for the Rio Grande do Sul State funding for science agency for paying the per diem here. That made it possible for me to travel 36 hours and be here with you all today. Well, as the title said, Baccharis is one of the most diverse genera of Astereae. It's maybe the 8th largest genera, or maybe the 4th largest genera, depending on the circumscription of some genera. And along with Mycarium, it was one of the genera of Astereae that lacks recent phylogenetic works. So when I was in São Paulo, I and a colleague of mine that was doing his PhD, Caetano, who did the work on Mycarium, and I was Baccharis, together with our advisor, we submitted a project to fill this gap in the knowledge of the most diverse genera of Astereae. So Baccharis is a genus originally restricted to the Americas. So it is found from northeastern Canada, eastern Canada to southern Tierra del Fuego in Argentina and Chile. And nowadays it is spread to Europe and Australia as well. In this area, the genus has several widespread species, like Baccharis salicifolia that occurs from eastern United States to Patagonia, but there are several other species that are narrowly endemic species that occur only in small areas of mountain summits. 90% of the diversity of the genus is restricted to South America. And in this area, there are 2 main diversity centers for Baccharis. One is the Andes, and the other one is southeastern South America. And the genus occurs in these 2 areas, mainly in grasslands, savannas, and mountain vegetations. In these areas, in the American continent, the genus occurs from mangroves and coastal areas in the lowland areas from the sea level, up to more than 5,000 meters high on the Andes. So it's a genus that occupies a very variable gradient of environments. And this reflects in a wide variety of habitats and forms of life, different features in several species. There are herbs, shrubs, subshrubs, trees, and even climbing species in tropical forests that are Baccharis. So the high number of species and the high variability of the genus made this like a taxonomic nightmare for identifications, because there is no one recent taxonomical work that covers all the area of the genus. Most of the keys that we have are for restricted countries or restricted areas around the American continent. And this genus is very useful. We see lots of works being done on Baccharis because it has economic importance. It's the source of several compounds for the pharmacy industry. It's a source of some teas, some beverages that are made of Baccharis, and also there are several species that are weeds and are toxic for the cattle. So there is a great economic importance on the genus. What makes Baccharis a unity? Well, most of the species of Baccharis, they have fused trichomes with a single adjoining basal cell. This is called the toothed indumentum of Baccharis, and the florets are always functionally unisexual. Baccharis was one of the first genera of Astereae that had a phylogenetic, molecular phylogenetic study. It was based on cpDNA RFLP markers, but this work was never published in a journal. So from this work, we see with the limited sampling that Baccharis was not confirmed as a monophyletic group. After the 90s, just recently, there was then the Compositae book, where Baccharis was treated only with few samples from just few species. And in this work, the Archibaccharis that was supposed to be the sister taxa of Baccharis was the sister of Baccharis. It was in a polytomy with Baccharis, along with other lineages that belongs to Baccharis. But the sampling in this work was very, very limited. only 1.5% of the species of the genus were sampled for the Astereae phylogeny. So the limited sampling that we had until nowadays did not provide satisfactory evidences about genetic relationships in the genus. So the proposal of this work is to provide a comprehensive phylogenetic hypothesis for Baccharis to test its monophily, clarify the phylogenetic relationships, and shed light on its evolutionary and biogeographical history. So I'm going to present these results in 4 parts. 3 of them are finished, and what is being done for the next parts of this work. For accomplish these goals, I did an extensive bibliography and a bio revision in South America, North America, and Europe, and then I could provide a checklist of 433 accepted species in the genus. Several field works were done from northwestern United States and northeastern United States to southern Argentina and Chile and Patagonia. I could gather samples from 13 countries. Some collaborators also sent me samples from other countries that I could not collect. And this resulted in more than 1,100 specimens of Baccharis collected, more than almost 700 silica gel samples. 15 new species were described, and 15 new species are to be described yet. And then I selected some of these samples to do the DNA extraction and sequence 4 regions for the phylogeny. And I could then have 248 species of Baccharis in the phylogeny, almost 60% of the genus, plus 44 outgroups of Astereae included in the analysis. All this data were analyzed by the means of Bayesian phylogenetic approaches. So this Baccharis monophyletic. Before, first of all, I did a phylogeny with all the data available on ITS and ETS to the tribe Astereae, and a limited sample of 57 species of Baccharis. In this analysis, yes, Baccharis is monophyletic. It's the red area highlighted in the phylogeny, and we could see which was probably the sister taxa of Baccharis, the sister clade of Baccharis. We have no idea about this before, and Baccharis is probably sister of the whole North American Astereae. This is a great news. And then these 2 clades, Baccharis in the North American Astereae, they are in a polytomy with several other South American lineages of the tribe Astereae. And the genus Archibaccharis is not closely related to Baccharis at all. So when you get a closer look to Baccharis in the phylogeny, where I put the 248 samples of different species, we see that smaller genera that were in the past segregated from Baccharis, they are embedded in Baccharis. For example, Heterothalmus, Baccharidioipsis, and Baccharidacea. They all are reversions to monoecious or other polygonal dioecious conditions, and are not perfectly dioecious, as are all the red species of Baccharis in this phylogeny. So we have to adopt a broad circumscription of Baccharis, or we have to segregate more genera from it. Well, when we look at the last proposals of segregation of Baccharis in smaller genera, none of them are monophyletic. There is a complete mess. And when we look in the proposal of infrageneric classification by Miller, it's not also giving us monophyletic infrageneric groups. So what could we do? We have to make Heterothalmus out of Baccharis. Several people in the southern cone, they love Heterothalmus because it's like a Baccharis that has like yellow ray florets that make it beautiful. But why to do that? Just because it has a yellow flower, and then what's next? You can split the Baccharis in 5, 6 genera, or maybe 10 genera, monophyletic genera. Well, they are monophyletic. They could be 22. They could be 33. We can create like 1 genera for every species. Is it useful? I think it's not useful because we have several species with economical importance. The classification was kind of stable along the time. And so I think we have to make it more simple to keep Baccharis in a broad sense that is just including more 5 taxa, because we have hundreds and hundreds and thousands of papers already published using these names. We have more than 11,000 species, for example, just in the Smithsonian Institution, named as Baccharis. Who would rename all of these? So just let's make the things easier. As it's written in this Bolivian chord, La Unión es la Fuerza. The union is the power. Let's keep Baccharis, all of them together. So we need to make it easier to deal with a big genus like this. So this is the reason to propose a phylogenetic classification. So I proposed 7 genera, subgenera, recircumscribed to reflect monophyletic taxa in Baccharis. For this, the subgenera Baccharis, Molina, and Trichonanthoides, they had to have new circumscriptions because the previous ones were not monophyletic. The subgenera Pteronioides and Stephananthus are embedded, and they are synonyms of the subgenus Molina. And 2 genera and 2 sections are moved to the level of subgenus to have all 7 monophyletic subgenera. So this is a closer view of the 3 subgenera Trichonanthoides, Obilongifolia, and Heterothalons. Here it's some samples of the subgenus Molina. It's a very variable subgenus. It's the second largest subgenus of Baccharis with about 200 species. It's very variable, and the sampling in the subgenus has to be improved in the next phylogeny. The subgenus Heterothalonolopsis has a single one species. It's a very interesting lineage because it has distinct features from all other species. The subgenus Coryndifolia was previously a section of subgenus Trichonanthoides, and now it's moved to the rank of subgenus since it's not closely related to any other subgenera. And then we have the subgenus Baccharis. The subgenus Baccharis is the largest one. It comprises half of the species of the genus. And for this subgenus, I could do a good sampling and prepare several new circumscriptions for the sections. So I recognized 17 sections within this subgenus. 5 of the previous recognized sections are synonyms, but these new sections, they have very distinct circumscriptions from the previous ones. For example, there are a group of species with winged stems in South America that are medicinal plants, that they were all placed in section Holopteria. And from the analysis, we can see that the winged stems evolved 2 times into sections that are not very close to each other. So this seems odd, since the winged stems is a very useful characteristic to classify the species. But then when you go to metabolomics, we did the metabolomics of all the samples that are in the phylogeny. We see that there are some chemical compounds that are, for example, defining every of these sections. This is very nice. It's going to be another paper that we're going to publish with the collaboration of the pharmaceutical sciences people. And there are also micromorphological characters that sustain all these new sections. So some sections now have broader circumscriptions as the section Axillaris. Here are some examples of this species in the section. And 2 new sections were recognized. One very interesting is section Andina from the northern Andes. No one before said that all these species were closely related, and they are all together, and they have some characteristics in common that are probably synapomorphic. So an outline of the results of the work. We have 433 species of Baccharis in 7 subgenera and 47 sections. The number of sections probably is going to be less in the next analysis because not all sections were already sampled. So for matter of stability, I did not make changes on the species from sections that were not sampled. And these infogenetic classifications gives the possibility to now revise smaller portions of Baccharis. And so I did the taxonomic revision of Baccharis subgenus Trichonanthoides, and I got a grant from Systematics Association to keep doing more taxonomic revisions of other 4 subgenera to advance so in the basic taxonomy of the genus that was, as I said before, a taxonomic nightmare. So here is the results of the taxonomic revisions of Baccharis. The first one, the taxonomic revision of the subgenus Trichonanthoides, is here. It's based on the phylogeny classification. It has 2 sections with synapomorphic characteristics. It was published last year in Phytotaxa. It's a group from southeastern Brazil, mainly. It's a classical taxonomical work with all the information, drawings, plates, maps that we need to identify and to know the species. And now I'm working on the other subgenera with this grant from Systematics Association on the taxonomy of subgenus Heterothalonolopsis, and to prepare a key to the subgenera on the revision of the subgenus Coryndifolia. That's a group from the southeastern South American grasslands. the revision of the taxon of the Baccharis subgenus Oblongifolia. That's a group restricted to the mountain summits across South America. And the taxonomy of Baccharis subgenus Heterodomus, that's a very intriguing group. And what's next from here and beyond? What I am planning to do? Well, with this phylogeny, we can answer some questions about Baccharis. For example, Miller proposed that Baccharis was originated in the Andes, just highlighting the Andean areas, Andean species in this phylogeny. We can see that the Andean species evolved independently several times. So the origin of the Baccharis of the genus is not in the Andes, but of some clades, yes, it is in the Andes, depending on which clade you are talking about. We can also explore about all the environments where the genus occurred, because there are several lineages that are restricted and diversified in different formations of the Atlantic forest, the savannas of Cerrado, the Harz with grasslands. So what's the role of niche conservatism or key innovations in this group? This has to be explored. Another thing that now can be explored with the genus is to investigate, to test hypotheses on the evolution of several vegetative traits.